I'd like now to introduce our webinar speaker, Dr. Renate Sekul. Renate received her PhD in biochemistry in 1989 from the University of Cologne, Germany. Since then, she worked in different pharmaceutical and biotech companies in the field of peptide and small molecule drug discovery in various indication areas. She is an expert in preclinical drug discovery, and her research fields comprise peptide synthesis and characterization, protein biochemistry, biochemical assay development, and profiling of drug candidates, as well as the development of array-based screening techniques. Renate has a profound expertise in array-based screening of pharmaceutical target proteins and antibodies using small molecule and peptide microarrays. She is the co-inventor and owner of several patents for peptide and small molecule drugs. Renate joined PEPperPRINT in 2012 as Head of Research and Development and is responsible for the exploration and implementation of new applications in peptide microarrays. So the subject of this presentation will be conformational epitope mapping on high-density peptide microarrays. So Renate, I'm handing the cursor over to you. Please take it away.
Okay, thank you, Carsten, for the introduction, and thank you everybody for joining our webcast. I will guide you to the presentation, and before I'm going to the more scientific part, I briefly introduce to you PEPperPRINT and its laser printing technology. I then give an overview on the principles of high-density epitope mapping. And finally, I will present our new application, which is conformational epitope mapping, and we have chosen for the case study the antibody antigen pair Rituximab and CD20. So PEPperPRINT was founded in 2011. It's located in Heidelberg, Germany, and it's a spin-off of the German Cancer Research Center. The PEPperPRINT founder have developed a proprietary technology platform for the synthesis of high-density peptide microarrays, which is based on a laser printing process. I'm going to explain a little bit later on. PEPperPRINT offers peptide microarrays for biomarker, autoimmune, and infectious disease research. We generate customized high-density peptide array and also we offer services in the field of epitope and biomarker discovery for academic researchers, for pharmaceuticals and biotech companies. We now really have a long-time research experience in all these fields, and we are continuously working on new application. So recently we have finalized a new application to address conformational epitopes. So how does laser printing or how does a laser printer works? We use a proprietary machine which allows to directly synthesize the peptides on functionalized glass slides. Instead of 4 color cartridges, which you know from a regular office laser printer, we use 20 cartridges which are filled with pre-activated amino acid toner. On the pictures on the right shows the laser printing machine, and on the bottom you can see the individual printing devices. For the first step of array production is the printing process. That means the individual amino acid toner are printed to the respective spot fields on the array, and the synthesis cycle is then initiated by a heating step to 90 degrees. Under these conditions, the toner melts and the pre-activated amino acids, which are needed for the synthesis, are released and are available for coupling. to the previous amino acid or to a linker for the first amino acid layer. A synthesis cycle is then completed by a washing step to remove excess material and by a deprotection step to cleave the N-alpha protecting group which is needed for the peptide synthesis. After this step, the array is then ready for the next synthesis cycle. The laser printing process has a couple of benefits. It's a very efficient and economic process because it works at very low material consumption. We have very fast production times, and the precision of the laser printing process allows to generate high spot densities with 800 peptides per square centimeter. We synthesize high peptide qualities because we are performing routine double couplings. One main advantage of the PEPperPRINT technology is its unique flexibility with respect to array content. Where conventional array production technologies works with pre-synthesized peptides, which are then spotted after synthesis completion to the microarray, we can directly synthesize the peptides on chip, which is a very high flexible process and which enables us to synthesize huge numbers of different peptides on the array, which is absolutely needed also for high-resolution epitope mapping, which is also very convenient to produce customized arrays at very low synthesis cost. We offer different array formats to meet the needs of different projects. So our standard format, which is shown here in the middle left, has a size of a microscopic slide, and it comprises up to 9,000 individual peptide features. A larger format, the discovery format, comprises up to 60,000 peptide features, and this is the format which is often used for biomarker and target binder discovery. Smaller peptide libraries can be synthesized in multiple copies on one chip, which allows to perform the parallel screening of multiple samples. And on the right you can see a microarray which comprises 16 small subarrays, and this is a very convenient format to set up assay conditions, and also it is very often used for hit validation. The PEPperCHIP peptide microarrays can be used for various applications. I have listed here some, and I will continue with high-resolution epitope mapping and conformational epitope mapping. So here you can see schematically how epitope mapping works. The first step involves the generation of a peptide library derived from the protein antigen of interest. The protein sequence is then translated into a set of overlapping peptides. We routinely synthesize 15 mer peptides with a maximum overlap of 14 amino acids. That means an offset of 1 amino acid. The workflow for the immunostaining is then comparable to any other immunoassays, such as an ELISA. That means we incubate our array with the sample antibody, and then we identify bound primary antibody with fluorescence-labeled secondary antibodies. After the immunostaining, we record the fluorescent intensity by microarray scanning, And finally, we analyze the data by an appropriate analysis software. We use the peptide analysis software for data analysis and for the identification of the underlying epitopes. On the left, you can see two examples of such an epitope mapping. The upper picture is an example, is a mapping of a monoclonal sample, and you can see one single stretch of amino acid. So, as expected, it recognizes a single epitope. The lower picture shows the mapping of a polyclonal sample. The red and green spots which are framing the arrays consists of control peptides we need for aligning the array. Here's a comparison of a standard epitope mapping and a high-resolution epitope mapping. So the difference between a standard mapping and a high-resolution mapping relies on the degree of peptide-peptide overlap. A maximum overlap with only one amino acid shift covers really all possible peptide motifs within a given protein sequence. So for the high-resolution epitope mapping, we generate sets of overlapping peptide with maximum overlap with an offset of one amino acid. In contrast, the conventional epitope mapping uses only partially overlapping peptides, typically with offsets of 4 to 5 amino acids. To illustrate how these differences can affect the results, we compared both approaches on a practical level. We performed for a protein antigen a high-resolution epitope mapping where we translate the antigen sequence in a set of 15 layer peptides with an overlap of 14 amino acids. And you can see the results here on the left. The microarray scan shows two adjacent but clearly separated signal stretches which translate into two epitopes of defined length. The same antigen mapped by a more conventional epitope mapping where we screened a peptide library with overlapping peptides with an offset of 4 amino acids. We obtained in the microarrays scan only one single epitope seen on the right picture, and also the detailed or careful data analysis could not ambiguously separate or resolve these epitopes as it was the case for the high-resolution mapping. I think this experiment demonstrates nicely that a high-resolution epitope mapping yields more comprehensive data, such as the clear separation of adjacent epitopes. You get precise information of the epitope length, and you can very precisely identify the conserved core motifs. More difficult to address are conformational epitopes. So conformational epitopes are hardly to identify on linear peptide arrays. Only in few cases I think we can be successful. We therefore developed constrained peptide libraries for conformational epitope discovery. Before I'm explaining in more detail the PEPperPRINT approach, I provide some information how linear and conformational epitope differ. So linear epitopes are composed of peptide fragments of contiguous amino acids, typically around 10 amino acid lengths, but some of them may be much smaller or also larger. The left picture shows an example of a linear epitope, how it binds to corresponding Fab fragment. It's a crystal structure of an Ebola peptide in it bound to the corresponding antibody. In yellow you can see here the extended conformation of this linear epitope, how it lies in the cleft of the antibody's binding pocket. Conformational epitopes are more complex and rely strongly on the protein's tertiary structure. So most conformational epitopes are discontinuous. That means that they are formed by amino acids or short peptide segments located on several stretches of the protein chain, and they are brought in proximity by protein folding. One example is shown here on the right picture. So this is a crystal structure of the HER2 protein to Herceptin, which is a therapeutic antibody which is used in breast cancer. The HER2 protein interacts, or the main contacts to the Herceptin antibodies are made by the yellow residues of the HER2 protein, which only comes into close contact upon protein folding. So most of the B-cell epitopes are conformational epitopes. Linear epitopes are T-cell epitopes, and some B-cell epitopes may be also linear. It is common knowledge that conformational epitopes can be mimicked by cyclic peptides. The idea behind this concept is that peptide cyclization may pre-organize and fix the peptide segment in a given conformation in order to mimic the proper structure of the protein antigen. Frequently, peptides are cyclized by disulfide formation between two cysteine thiols. For some reasons, I will explain them in a second, we apply a different cyclization strategy. We developed a side chain to N-terminus cyclization between a C-terminal cysteine and the N-terminus of the peptide. This results in a stable thioether linkage between the thiol and the N-terminus of the peptide motif. The advantage is the resulting thioether linkage is chemically very stable, and it's also compatible with any other amino acid in this peptide sequence. We didn't observe any side reactions. For conformational epitope mapping, we also synthesize our peptide loops in 3 loop sizes, small, medium, large, to cover different conformational constraints. For our case study, for our proof of concept study, we have chosen as model the interaction between Rituximab and CD20. The protein antigen CD20 is a transmembrane protein with 4 transmembrane segments, which connect the cytosolic domains and also the extracellular loops. CD20 has 2 extracellular loops. One is very small with a length of 8 amino acids, and it is located close to the membrane. One is much larger with 40 amino acids, and this loop is also structurized by a disulfide bridge between cysteine 167 and cysteine 183. The Rituximab antibody is a chimeric monoclonal antibody, and early mutagenesis studies and later on co-crystal structure have shown that Rituximab is directed against the extracellular loop of the CD20 protein. Rituximab is used for the treatment of B-cell lymphoma and of autoimmune diseases such as rheumatoid arthritis, and it's on the market, I think, at least for 15 years. It is also known that reduced, denatured CD20 is not recognized by Rituximab, so clearly there's a high conformational influence on antibody recognition. And this was also confirmed by a co-crystal structure between this part of the CD20 loop and a Fab fragment of Rituximab. And here you can see the binding mode and also the conformation the epitope peptide takes in the crystal structure. So it's a loop structure, and the main contact are made by a few amino acids which are located in this part of the molecule, which is also deeply immersed in the Rituximab binding pocket. So we started our proof of concept study with Rituximab. We created we synthesized two microarrays with linear and cyclic peptides derived from the CD20 extracellular loop. This sequence was translated into a set of overlapping peptides, and we have chosen peptide lengths of 7, 10, and 13 amino acids. We have also chosen for high resolution mapping a peptide-peptide overlap, maximum peptide-peptide overlap with an offset of 1 amino acid. Here you can see the microarray scan of the linear peptide library. We didn't observe any signals after incubation with Rituximab in staining, so Rituximab didn't recognize the linear peptides. The picture was completely different with the constrained peptide libraries. Here, this picture shows the microarray scan on the constrained peptide libraries, and we observed clearly resolved and very strong signal pattern with highest intensities with the 10 and with the 13 mer peptides. Data analysis illustrated also by this intensity plot gives you a more quantitative picture. So the intensity plot is a plot for the generation of an intensity plot. The fluorescence intensities are plotted against the peptides on the array. We observed only low signals with 7 mer peptides, but much higher signals between towards the 10 mer loops and also the 13 mer loops. A detailed data analysis shows then consensus as epitope, a consensus motif of 6 amino acids starting from glutamate 168 to serine 173 as Rituximab epitope. To characterize this interaction and the epitope in more detail, we furthermore performed a substitution analysis. A substitution analysis is a very useful tool to determine precisely the conserved and variable amino acids within a given epitope. For a substitution analysis, each amino acid position within the wild-type epitope is substituted by all 20 physiological amino acids. In principle, the substitution analysis is comparable to an alanine scan. However, not only alanine the positions are not only substituted by alanine, but by all other 20 amino acids in each position. The starting epitope we used was the top hit or the Rituximab epitope from the 13 mer library, and then synthesized or generate a substitution library out of this motif, and each of these peptides were printed in triplicates as single spot subarrays, and this is also reflected in this microarray picture which we obtained after the immunostaining with Rituximab. So each row in this on this microarray represents one amino acid position in the peptide library, and each column represents the amino acid replacement, starting with alanine on the left and ending with tyrosine on the right. And you can also see from the primary scan that some parts of the Rituximab epitopes are highly conserved, whereas another part seems to be highly variable. A more precise picture is given in the heat map, which displays the averaged spot intensities of each amino acid replacement. So the sequence is plotted here from the C-terminal part of the epitope to the N-terminal part, and from left to right are the amino acid replacements. So you can see that the C-terminal part is the most conserved part of the epitope, and the N-terminal part is highly flexible. For the most comprehensive picture, we generated then an amino acid plot. This plots the wild-type epitope intensity, or this sets the wild-type epitope intensity to 100%. This is the wild-type epitope. And all other amino acids replacements are then spotted as relative intensities to the wild-type epitope. The sequence here is plotted from the N-terminus to the C-terminus, and as already visible in the microarray scan, the first 5 N-terminal amino acids are highly variable. Many amino acid replacements are well tolerated or yield even higher values, but the final 8 C-terminal amino acids are highly conserved. The highest degree of conservation showed this peptide stretch from NPS. So these residues were absolutely conserved, and no other amino acids were tolerated in this position. Also, a high degree of conservation was observed with alanine. So in this position, only 2 conservative replacements by cysteine serine were tolerated, whereas the other amino acids in the Rituximab epitope showed a little bit more flexibility. Our data are in coincidence with the crystal structure data, where the tetrapeptide motif ANPS was located in the part of the conformational epitope which was deeply immersed in the Rituximab binding site and which made the most contact also to Rituximab. However, we could identify these preferences more rapidly than to generate co-crystals and get co-crystal structures, so we can perform these experiments very rapidly and also in a very fast fashion. So to summarize, the substitution scans allows precise epitope mapping and analysis of therapeutic antibodies. It's very useful also for cross-reactivity analysis. It's very useful also for patient stratification upon mutation, upon analysis of protein mutations, and it allows also the comparative analysis of biosimilars to a one residue resolution. I think the key features of our conformational epitope mapping approach are that our constrained peptide libraries are based on cyclic peptide thioethers. The cyclization chemistry is compatible with all amino acids. We didn't observe any side reactions. We think it makes absolutely sense to present constrained peptide libraries at different loop sizes to cover different conformational constraints and also to mimic several conformations among or within a protein antigen. You get a detailed view on variable and conserved amino acid of an epitope, and if you have identified the epitope, you can run substitution scans which give you even more detailed information about conserved and variable amino acids. So if you are interested in the PEPperPRINT technology, we have on our websites some tutorial videos and webinars, and also some application notes also on the case study I presented. So thank you for your attention, and yeah, I'm ready to answer questions.
Okay, great. Thank you very much, Renate. So again, if you have any questions, please use the questions pane and type your question, and I will read it to Renate. So here's actually one, Renate.
Yeah.
Someone from the audience is asking, does the method also work for discontinuous conformational epitopes? Can you briefly comment on that?
Yeah, I can say that it works also with discontinuous epitopes. We are currently working on a new case study, and the first data are very, very promising, and I think we will publish them soon. But I think I know that discontinuous epitopes are even more difficult to address than continuous conformational epitopes, because each loop makes a contribution to the binding affinity of an antibody, and I think also one prerequisite, I think, to identify or to map continuous epitope is that you have a minimum affinity. But normally discontinuous epitopes are B-cell epitopes, which makes also more contact to the antibody. That means discontinuous epitopes are larger and more residues are making contact also to the antibodies. So I think we have a good chance to map discontinuous epitopes, but however, it's very clear, I think, that we will also miss some epitopes.
Great, thank you, Renate. So another question here. What is the advantage of the thioether linkage compared to disulfides?
I think if you synthesize peptide disulfides, you have it's an oxidation process. I think there are many reagents then for the preparation of disulfide bridges. The problem is that you have with all reagents oxidative byproducts. That means it's not very well compatible with other cysteines within the sequence. You have to replace them. Also, these cysteines can form other disulfide bridges. And also we have other oxidation-sensitive amino acids in a peptide. For example, methionine can be oxidized under cyclization conditions to methionine sulfoxide, or also tryptophan is known to be oxidation sensitive. So I think the thioether linkage or preparation of thioether is in this sense our preferred method, because we didn't observe any byproducts. It's also a very stable bond, and we will not only use this for conformational epitope mapping, but it's also interesting for target binder discovery, where you also work with other protein targets than antibodies. And with thioether peptides, you have also more flexibility in these cases in the selection of your buffers. For instance, you can add DTT or TSAP or whatever, which is not compatible with disulfides.
Okay. Thank you, Renate. Another question here is, in the constrained cyclic peptides, how to verify monomer cyclics instead of oligomers that form in the high-density peptide spot?
We have done the setup of our cyclization chemistry. We have developed them on solid phase with a cleavable linker. That means we have really optimized to get to get cyclic monomers and not cyclic dimers or whatever. And we tried at least 100 different conditions to synthesize our cyclic thioether, and this development or protocol development was always controlled with analytical techniques like mass spectroscopy to verify that we get the monomeric thioether. That means we synthesized, or we developed on solid phase with a cleavable linker, and we tested with a couple of model peptides, not only with a single model peptide, but with a couple of model peptides. After running the cyclization, we cleaved off the peptide from the solid support and did a mass analysis. And then we were able, after a couple of months development, to find out optimum conditions. However, I think maybe we have, under array conditions, a few side reactions, but I'm very confident that the main product on our arrays is really the monomeric cyclic thioether.
Okay, thanks, Renate. Another question here from the audience. What are the minimum and maximum lengths of epitopes that you have so far mapped with cyclic peptides? Can you comment on that?
I think the minimum, because we haven't generate not hundreds of case studies, so and typically I have to say the minimum ring size we will synthesize on the array is a 7-mer. We don't synthesize smaller rings because in this case, very small rings have a high ring constraint, and I think the efficiency of the cyclization process will not be good. Therefore, I think the minimum loop size we offer is a 7-mer loop size. In internal projects where we analyzed and looked for conformational epitopes, we identified consensus motifs of 5 to 8 amino acid, I would say. And I think if you find them among your cyclic peptide motifs, if you find, for instance, 7-mer motifs, then it's also crystal clear that probably not all amino acids within these cyclic motifs are making contact to the antibody, but there are for sure the hotspots of interaction.
Okay, thanks, Renate. Hold on, the questions are rolling in. Thank you very much to the audience for your interest. Let me here find another one. I'm trying to read them in the order as they appeared. Someone from the audience asks, please let me know in case of glycan epitopes how this technology would be used.
I'm not sure if I have understand this question, so not acoustically, but could you please repeat it?
Yeah, it's. I think the reader is interested in glycan epitopes.
Yeah.
GLYCAN.
So,
and how this technology of conformational epitope mapping would be used or applied. I mean, in case you may be familiar with that, Renata, otherwise we'll skip the question. And then,
if I understand rightly, the question is if we can map glycosylated conformational epitope, or so otherwise I think we can think about it, or maybe the person interested in this topic can maybe more, I would say, more precisely address this question, and we can answer them later on.
Okay, good. So let's maybe the person who posed this question can elaborate a little bit more on the details of the questions. And again, we will be in touch then.
Okay.
And I think I can almost tell that we won't be able to address all the questions, and we also limit the time of the discussion about 20 minutes, otherwise it just gets too long. But we will address all the questions offline and email the individuals back. Here's another question perhaps. Are there any limitations or difficulties seen with the antibodies, for instance, IgM, mouse, IgG1, used to map? So are there any limitations or difficulties seen with the antibodies used to map? I'm just reading it word by word here. Does this sort of make sense to you, Renate?
I have to say we didn't map the conformational, or we didn't test IgM antibodies with our constrained peptide libraries. I think we test a lot of IgM responses with our linear peptide epitopes. This works very well, but so far we never tested IgM on our constrained peptide libraries. For our case studies, we use therapeutic monoclonal antibodies to validate our technology, and I think they are mainly IgG1 and IgG2.
Okay. Somebody here was also interested in the turnaround time for linear and conformational epitope mapping. Could you briefly elaborate how long it may take for our listeners?
Yeah, I think for linear epitopes, I think we offer them, I guess, in 6 to 8 weeks, and for conformational, we offer them for in a time frame of 10 to 12 weeks. We are doing a lot of projects parallel at PEPperPRINT, so I think to be on the safe side, or to plan conservatively, I would say for the constrained peptide libraries we need 12 weeks, but we always try to deliver earlier.
Okay, great. Here another question that's coming up. Have you screened these chips for cell binding instead of protein or antibody binding?
No, we didn't screen our chips with cell suspensions.
Mm-hmm. Okay. I'm still scrolling through a little bit through the different questions here. I think here's one for true conformational epitope. For a true conformational epitope study, one would need a much longer peptide. What would be the possible maximum length in the future? Or sort of what kind of peptide lengths we may be able to manage in the foreseeable future?
I think for the sake of quality, typically we synthesize 15, 70 mer peptides. For some projects, we synthesize also 20 mer peptides, but we don't offer longer peptides.
Mm-hmm. Okay, thank you, Renate.
Okay.
Maybe I'll read one more question here by someone in the audience. The question is there a way to discriminate between amino acids directly involved in the interaction with CDR or amino acids involved only in the epitope conformation? So I can read that again. Is there a way to discriminate between amino acids directly involved in the interaction with CDR or amino acids involved only in the epitope conformation?
I think partially the question maybe can be solved, I think, by a substitution scan, but I think we have to think in detail about this question and to discuss this internally, how to run such a project or how to perform these, or to answer this question on an experimental level.
Okay, great. So, well, thank you, Renata. I think I'm going to conclude it here. So once again, if your questions haven't been read and answered, we will answer you offline by email and get back to you. So definitely no question will remain unanswered. So I'm going to conclude this webinar presentation. Again, thank you all for attending this webinar. And we also would like to thank our speaker, Dr. Renate Sekul. We will make the presentation slides and the webinar file available online on the PEPperPRINT website within a couple of days. So you can visit the PEPperPRINT website and download the webinar file for further listening. And again, if you have any questions further on, please contact the PEPperPRINT team directly. You can go through the website or the different contact informations that you may have. Please also check out our website for updates, future events, or webinar announcements. And so I will conclude and end this webinar now. Thanks all for dialing in, and we look forward to welcome you our next presentation. Thank you very much, and goodbye.
Bye-bye.